Negat.
Oh, okay.
It's nice.
Yeah. Yeah. It's a nice idea. Like webinars. Yeah.
Yeah. Kaushik, should we start? Okay, let's start. So let me introduce our speaker today. So this is Dr. Path Rakesh Desai. He is a former lab mate of me. He finished his PhD from University of Maryland. His PhD was checking the effect of base pair mismatch on DNA plectonome. He worked on University of Maryland under Dr. Siddhartha Das and in collaboration in National Institute of Health under Dr. Kiersey Neumann. Now he is a postdoctoral researcher at Northwestern University and he works with John F. Marco. He has lots of experience on simulating polymer DNA and also lots of experience on experimental DNA research, single DNA. So I think we are up for a very good seminar today. Welcome, Path.
Yeah. Thank you. Thank you for the kind introduction. Should we get started or do you want to wait a few minutes for people to join in?
I think we can start slowly.
Oh, okay. I can start if you. All right. So good morning, good night, good evening, everyone. I don't know what time it is there, but so today I'll be presenting some of my thesis work on trying to understand how defects affect DNA mechanics. Right. And before I get into that, I just want to like introduce some of my previous work that I did for my master's, where I was trying to look at how polymers behave in confinement. And then I can like show you how that connected to my DNA work and how I got actually started with the NIH project. Right. So when I joined my Dr. Das's lab for my master's, I was working on how polymer grafted surfaces interact with each other. Right. So we are trying to look at this compression regime where the polymers aren't like completely compressed. They're like almost halfway compressed, right? So you have some polymers that would be like in the stretch mode, and then some that would be in the compressed mode. And then we are trying to understand how that affects the shearing behavior and interactions between these two grafted polymers. And then another project I worked on, which was also very similar in terms of the methodology, was this how soft polymeric drops interact with soft polymeric surfaces. So usually you would have like a soft polymer surface, but then you would have a nanoparticle, rigid nanoparticle interacting with that surface, or you would have like a hard Leonard-Jones surface, but then you would have like a soft polymer particles. But we are trying to understand how soft polymer particles behave on a soft polymer surface. And both of these worked use the classical bead spring model, which I hope you are familiar with. I won't be talking much about it. I just wanted to bring it up to show how I got started with the research. And yeah, so the most of my master's work was done on these two topics. And again, they might be separate areas of research, but they use like same methodology. So it was very easy to transfer skills from one project to the other one. And then over here, we kind of looked at what's the effect of how the surface hardness on the polymer behavior. And then our findings had some interesting implications for self-lubricating surfaces. But again, I won't be talking much about that. So today, mostly I'll be talking about my thesis work, which was on DNA mechanics, right? So DNA, for a brief introduction, this classic helical structure. So it's almost like a twisted ladder, and then each strung of the ladder has what are called nucleotides, right? So that each of these single unit would be called a nucleotide, and they both together make what's called a base pair. So each rung of this ladder is a base pair, and then the individual components are nucleotides, right? And then DNA, if you know, has this very 4 specific type of molecules that it's made of, right? So there's adenine, guanine, cytosine, and thymine. And for like shortly, they are called A, T, G, C. So just by the first name. So the structure of DNA is pretty complicated, but very simplistically, you have this backbone, phosphate backbone, that connects the 2 base pairs together. So that's a region here. And then you have a sugar group that's connected to the phosphate backbone. So the phosphate backbone and the sugar group are same for all the molecules, right? But then at the end, you have what's called bases. So these are small compounds that have a specific structure. And then this specific structure helps them to form what's called hydrogen bonds. So those are non-covalent bonds between an opposing structure. So this is one of the ways where your body knows what base pair goes with what. So here A will form a very good two good hydrogen bonds with T, and then G will very specifically form 3 hydrogen bond with C. And then they usually, so A and T go together and G and C go together. But my focus was trying to understand what happens if, all right, one of them is misplaced. So it's either GG or AA or something like that. How does that affect DNA structure? And then these mismatches are kind of important in your body, because when you have a mismatch, and if it's not corrected over generations, as your body self-replicate, that mismatch will keep on aggregating more defects next to it, right? So that's one of the reasons why you have like the hereditary diseases, where the not just the mismatch, but other defects are also not corrected by your cells. So we're trying to understand how these defects affect the DNA structure, and then can that effect somehow give us a clue on how the protein actually corrected. Right. So briefly, to simulate DNA, you have a lot of different models available to you. So you can start from the smallest, what's called quantum scale, where you simulate all the electron waves and stuff like that, where you can simulate maybe 1 or 2 nucleotides. And then you can have like a femtosecond long simulation. And then going on to a longer scale, if you want to study projects that, if you want to study phenomenon that take a longer time, you have the all atom simulations where you individually simulate each atom, but you don't simulate the electrons. So each atom you represent as a spherical molecule, and then you like approximate the interactions between those spherical molecules. And then over here, you can have like up to 200, 300 base pairs, and then you can have like maybe a microsecond long simulation, but that takes a lot of computational resource. The main focus, my main focus was this coarse-grained regime. So coarse-grained is when you combine two or more atoms together as a single unit, and then you approximate the forces that are applied on that single unit. And so there are varying level of coarse graining that people can do specifically for DNA. You can have this level of coarse graining where each nucleotide is kind of a single atom instead of a project of multiple atoms, or you can have this even coarser grain where you have multiple base pairs that are approximated as a single bead, and then they are connected by a spring. So this is where my connection to my previous work came in, where you can also simulate DNA through the bead spring model. And that was kind of one of the first projects we thought could be interesting on that scale. And over here, you can go up to like hundreds of microseconds for this level of coarse graining. And then for even more coarser graining, where you use the bead spring model, you can simulate DNA plecton. You can simulate DNA for seconds. So those would be the experimental timescales where people have seen things over seconds, stuff like that. And then if you want to go into the theoretical work, you can use what's called the worm-like chain model. Again, not going to go into detail into that, but it's also very similar to the polymer worm-like chain model. And then you can come up with theories to explain the experimental phenomenon using this mean field model, right? So the first question we are interested in is what happened? How does DNA buckle at the defect? So by buckling, I mean, if you have like a garden hose that has a kink in it, so that's like a very strong band. And if you twist it, right, it'll form this buccal structure where it's like self-crossing, and it'll always form this buccal structure at that kink. Right, and this is true for any wire. So even like any wire you have now, if you try to twist it up, it'll form this buccal structure. And then if you have a kink in there, it'll form the buccal structure at the kink. So we wanted to understand, all right, would the same thing happen to DNA? Right, so maybe on the micro scale, you have this mismatches that are softer than the rest of the DNA, right? They're weaker. So would the DNA, when you twist it, like buckle at the mismatch? And then this could be really important in this mismatch recognition, because if you look at the amount of DNA in your cells, it's a lot. So if the mismatch recognition protein has to go along every single base pair to find a single mismatch, it would be really inefficient. It would take a long time, right? So we were thinking that if the DNA could buckle at the mismatch, again, because it's like weaker at the mismatch, then you have this kink structure where the DNA could buckle. So if the DNA buckles at the mismatch, this mismatch would be localized at these ends here, where you have these loops, right? So not only now the protein has to find less, protein has to test less of the DNA, but then it's also like conveniently located at the ends where it's more accessible than in the middle of this mess. And so that was our thinking going into the project, to try to understand how mismatches affect the DNA plectoneme DNA buckling, and then could that give us an idea of could this lead to mismatch recognition? Right. So briefly, a presentation outline. So today I'll be presenting some of the experimental work my colleague did before my simulation work, and then I'll present some of my simulation work and then I compared my simulation work to the existing experimental work, and we found that it compares very well. And then finally, I kind of extended my simulation work to a negatively supercoiled regime. So I'll go over what negative supercoiling is and why it's important later on. But so this is a brief outline of the presentation. Right. So before I go into the results, I wanted to share a little bit about what's called DNA topology. So DNA topology is basically trying to understand what the structure of DNA is. And more specifically, it's trying to understand how many times the DNA crosses itself, how many times the DNA helix crosses itself. So the DNA topology structure defined by the linking number is divided into two parts. So one is the twist. So that's how many times the two strands of the DNA are intertwined with itself. So if you think of the ladder, if you think of a helical ladder, you're thinking of how many times the axis of that helical ladder is twisted, right? And then the rise is how many times of the DNA helical axis crosses itself. So it'll be more clear when I show some figures, right? So in a relaxed DNA, you have this double two-strand structure, it's called a double-stranded DNA, an intact DNA, right? And then the two strands are intertwined with each other through some twist. So that's we call twist 0. Again, these numbers are not important. It's just to give you an idea of what we mean by saying relaxed and supercoiled DNA, right? So it's DNA has some inherent twist, which is twist 0 or relaxed twist, right? And then when you have a relaxed DNA, you can see that the two strands of the DNA are crossing each other. So that's twist. But then the relaxed DNA helical itself is not crossing itself. So the rise is 0 right now. Right? So there is some inherent twist in DNA, but there is no rise because the helical axis isn't crossing itself. Right. So now, because DNA has a direction, so it's a right-handed helix. If you apply torsional stress or if you twist the DNA in the direction of the helix, you overwind the DNA. So when you overwind the DNA, the twist obviously increases because you're overwinding. And then if it buckles and forms this what's called a supercoiled structure, supercoiled rise structure, right? You will have this crossing, and then that's defined as a positive rise. So positive and negative rise aren't really that different. It's just semantics, right? So if you apply overwinding, you get positive rise, and then if you apply twist in the direction opposite to the helical axis, right? You reduce the twist of the DNA, and then again when it supercoils, you have this negative rise. And over here, it's really interesting because if the DNA didn't have this right-handedness, if you over twist or under twist it, it would have the same effect. But because the DNA axis, the helical axis has this handedness, if you under twist it, what happens is you make the base pairs, so the like individual structure, weaker, and this makes the whole DNA weaker. So this has some interesting implications later on, but this is something to keep in mind, that when you overwind the DNA, the base pairs interactions like aren't affected that much as when you underwind the DNA, when you're really weakening the structure of the DNA. And then finally, one other thing that I would like to introduce is called the specific linking number. So it's basically a way to compare the DNA topology of DNA of two different lengths. So if you're trying to compare like DNA of, let's say, 10,000 base pairs to DNA of 1,000 base pairs, the idea is that the one turn of twist you apply to 1,000 base pair DNA and 10 turns of twist you apply to the 10,000 base pair DNA should produce similar effect in the DNA, right? So that's where the specific linking number comes in, because we have like simulation, theoretical, and experimental work. It's really hard to get the correct DNA length scales for all those three, right, to compare them directly. So that's where the specific linking number comes in, where we can use this to compare DNA from simulation to experiments, where the length scale are obviously very different, right? So next, before I introduce the experimental work of my colleague, let me briefly go over the experimental method that is called a single molecule magnetic tweezer, right? So very simplistically, in this method, you have an objective, and then you have a cover slip, right? And then on the cover slip, you attach one end of the DNA. So this is a single DNA molecule, right? So, and then on the other end of the DNA, you attach a magnetic bead. So when you have magnets close to the magnetic bead, you can apply force to the magnetic bead, obviously, because it's magnetic. And then you can also apply rotations by rotating the magnet, right? So because this one end is both ends are fixed, the DNA is called torsionally constrained. So the only way it can relieve the torsional stress is through buckling, right? So when you twist the DNA using these rotations, right, it'll absorb the twist for some time, and then when it can no longer absorb the twist, it will buckle and form this supercoil, what's called the plectoneme structure, right? So it'll form one loop. So it'll form the first loop when it can no longer absorb the twist, and then as you apply additional turns, the plectoneme loops gets bigger and bigger. Right. So in practice, what this looks like is when you apply turns. So it's again over here a changing linking number. As you apply the turns, let's say for 0.2 piconewtons of force. So this is kind of one of the lower forces people do for single molecule experiments. Right, so for 0.2 piconewtons, as you apply the turns, the DNA absorbs this twist until some point, right? So it's still around the same length as when it was relaxed. So it's just absorbing the twist you're applying. But then when it can no longer absorb this twist, it buckles and forms this supercoiled, this first plectoneme loop. And over there, you see a sudden decrease in DNA length when it forms this first plectoneme, DNA extension, I should say, when it forms this first plectoneme loop. And now as you apply additional turns, all of those turns go into elongating this plectoneme loop. So now every turn results in like a uniform decrease in length. So you can see that's a straight line as you apply more turns. And because it's kind of 0.2, it's kind of one of the lower forces, the DNA structure isn't weakened a lot. So positive supercoiling, so overwinding, and negative supercoiling and underwinding have the same effect, right? So you have this symmetrical curve where the DNA structure doesn't play much of a role. It's all like DNA, it's all polymer physics almost, where the DNA buckles without any respect for the base pairs. Now, if you go to a higher force, let's say 2 piconewtons, over there for the positive, again, you see a similar thing where when you apply overwinding, you see this, the DNA absorbs the twist, so there is no decrease in DNA extension. And then when once it buckles, it forms this plectoneme structure. So you have this sudden decrease in DNA extension. And now as you apply additional turns, the DNA extension keeps decreasing because each turn just elongates the plectoneme and then there's like less DNA length. But now when you look at this high force at the negative supercoiling case, right, you can see that throughout, as no matter how many turns you apply, the DNA doesn't buckle, right? So over here, the base pair effects come into play, where, like I mentioned before, negative supercoiling, you are kind of weakening the base pairs. So what happens is when you have high force, you weaken the base pairs. Now, as you apply additional turns, the DNA doesn't actually have to buckle. It can absorb those turns by melting at some point along the DNA backbone, right? So it no longer has to buckle. It can just absorb the turn through melting somewhere. And so now you can see that there is no reduction in length as we apply more negative turns, because it's just locally melting instead of buckling and forming a supercoil. Right. So, and then one of the other important thing to mention is when you have this only extension data, you do not know where the plectoneme has formed along the DNA backbone, right? So this plectoneme, whether it's in the center or at close to the surface or close to the bead, you get the same data. So you don't know where the plectoneme is formed. It's just something to keep in mind for later on, right? So one of my colleagues, Dr. Dittmore, he was a postdoc in Dr. Newman's lab. He wanted an experimental way to try to understand, does DNA buckle at the mismatch? So he did this magnetic tweezer assay, but instead of having the mismatch in the center of the DNA, he had the mismatch very close to the surface, right? So now as you apply the turns, the DNA will absorb the twist again. And then once it can no longer absorb that additional twist, it will buckle to form the plectoneme structure. And then you have this sudden decrease in length. And now as you apply more turns, the plectoneme structure elongates, right? But now because the mismatch, that's the red dot here, was very close to the surface, what happens is as the plectoneme elongates, it takes length from both above the mismatch and below the mismatch. But because below the mismatch, it's very close to the surface, there is a limit on how much it can elongate, because like, again, once it's at the surface, there's nothing to absorb from below, right? So over here, by having the mismatch close to the surface, he could look at plectoneme localization at the mismatch by trying to understand this free buckling signal. So what's happening here is once the plectoneme is long enough that it can no longer elongate, it interacts with the surface. And now as you apply additional turns, because the plectoneme can no longer elongate, you're again in a state where the DNA tries to absorb those turns through twist, right? And then it can absorb some of those turns to twist, but then once it can no longer absorb turns, you see this second buckling transition. So you form a second plectoneme too, right? And that's when, and then again, as you apply more turns, the plectoneme keeps on decreasing. The DNA extension keeps on decreasing. And this is how he was able to infer if the plectoneme was actually pinned at the mismatch by looking for this rebuckling transition, right? And one of the limitations here is the plectoneme always has to stay pinned at the mismatch for him to see this rebuckling signal. Because even if it's like, let's say that 99% of the time, he won't be able to see this rebuckling signal because the DNA would just move, because the plectoneme would just move along the DNA backbone. So the plectoneme has to stay pinned at the mismatch 100% of the time. Right. And then to explain this kind of effect where he saw that a single base pair mismatches in a 6,000 base pair DNA, so like a very small mismatch, can cause this plectoneme localization at the mismatch, right? A single mismatch. So to explain this, we collaborated with Dr. Marco's lab at Northwestern, and they came up with a theoretical model of why this plectoneme pinning actually happens. And I won't go into much detail about that, but very simplistically speaking, this plectoneme pinning is a result of competition between two forces. So because it's kind of the DNA mismatches are weaker than the intact DNA, it's easier to bend at the mismatch, right? So the plectoneme for the DNA would like to buckle at the mismatch because it'll be easier to form this bend because the DNA there is weaker than the DNA along the rest of the molecule. But then if you form the plectoneme and if you stay pinned at the mismatch, you can no longer move up and down along the DNA backbone. So you lose that entropic energy of that motion, right? So the plectoneme pinning actually, according to that theory, is a competition between this ease of bending at the mismatch and then the entropic loss that you get by the plectoneme staying pinned there. And to test this model, Dr. Dittmore did some more experiments where he varied the salt and force that was applied to DNA. So salt and force have somewhat of a similar effect. So as you increase the salt concentration, because the DNA backbone is negatively charged, you screen more of the negative charge as you increase the salt concentration. So at higher salt concentration, the DNA becomes easier to bend because the negative charge is screened. So there's like no repulsion here at a very strong bend, right? And then at a higher force, when you form the first initial loop, you're kind of pulling against the force, right? So at higher force, you have to pull more. So you would want the plectoneme, you will want the end loop to be as small as possible so that you have the highest length possible. So as you increase the force and you salt, you have this stronger bend in the DNA. And then according to that model, that stronger bend should result in more pinning at the mismatch. And in the experiments, Dr. Dittmore found that actually did happen, that as you increase the as you increase the force and the salt concentration, you observe more pinning for higher forces and salt concentration. But again, because he was limited by not having direct data of where plectoneme pinning was occurring, but just being able to infer it from the classical magnetic tweezer experiments, he couldn't look at what happened in like a physiological regime. So physiologically in your body, the salt concentration is around like 0.2 nanomolar salt, 0.2 micromolar salt, right? And then the force is actually acting on your DNA as typically much longer. So because of the experimental limitation, we couldn't study what happened like physiological condition of like lower salt concentration. And so my project was to come up with a simulation framework where we could study what happens to the DNA under physiological condition. And not only that, but also extend that model to study negatively supercoiled DNA. So in your body, DNA is always negatively supercoiled, stored as a negatively supercoiled DNA. And as I mentioned, the negative supercoiling weakens the DNA base pairs itself. And if you have a mismatch, the negative supercoiling will weaken it even further. And so that weakening somehow enhance the plectoneme pinning at the mismatch. So those are all questions we wanted to answer with my simulation work. So briefly, to do this simulation work, I chose what's called an OxDNA2 model. So it's an Oxford DNA. It was made at Oxford, made at a lab in Oxford. So that's why it's called like Oxford DNA, OxDNA, right? And then it's a coarse-grained model where the base pairs of DNA are approximated as rigid nucleotides. So sorry, the nucleotide is approximated to the rigid ellipse, right? So over here you have, let's say, like 80 atoms in each base pair. This model reduces those 80 atoms to 2 atoms, so 2 rigid ellipsoids with 3 different interaction sites for the phosphate backbone, for the sugar group, and then for the base itself. Right, so you're dramatically reducing the number of interactions you have to calculate, so you can go to like much longer time scale. And then in this model, you have like two rigid nucleotides that are simulated as ellipses with 3 different interactions, right? And then the model has like salt dependence and stacking interaction, stuff like that. Again, not going into detail there, but it's been well tested against like single molecule experiments, right? And it's shown that it can compare favorably to single molecule experiments. So that's why we kind of started with this, using this. Right. So the first question we had was, can you even use this model to study what we wanted to study? That was mismatches, right? So because our theories told us that the plectoneme pinning was actually a result of this ease of bending at the mismatch, first we wanted to see, can OxDNA reproduce the bending rigidity of a mismatch and how that compares to the all atom simulation. So over here, we're assuming that all atom simulations are kind of the gold standard for simulations, right? So if OxDNA can be close to that, we can be confident that it will give us like quantitative answers to our questions, right? So the first thing I did was I had like a short DNA molecule, and then I used umbrella sampling to bend the short DNA molecule from a straight DNA, so that's like 180 degrees, to a very strong bend, right? And then I compared that bending energy that I got from the simulations to the already published data for all atom simulations. And I could see that the OxDNA was pretty close, OxDNA results were pretty close to the all atom simulations, right? So you can see over here, there's like 2-3 kts of difference, right? So it's not quantitatively accurate, but it's close enough that we can use it, right? And then, so that's why we are confident we can use the OxDNA model to study this plectoneme pinning, because it can reproduce this bending rigidity at the caused by the mismatch. And then next we wanted to see what happens when you have more mismatches. How does that affect the bending energy, right? So over here in this simulation, we have like a 30 base pair DNA molecule, and then we introduce 2, 4, and 6 mismatches at the center of the molecule, and then again we bend the molecule, and then you can see as you bend the molecule, you see that you need more and more energy to bend the DNA as like to a more degree, because the DNA is negatively charged. So you need like a lot more energy to overcome that negative charge repulsion, right? And then you can see as you increase the number of mismatches, the DNA becomes even softer. So the mismatches add up, right? So for 2 mismatches, you have some reduction in the bending energy, but then for 4 and 6 mismatches, the bending energy is reduced even more. As you increase the number of mismatches, the DNA becomes weaker and it becomes easier to bend to a particular degree. Over here, another interesting thing to look at is that this effect of mismatch is actually non-linear, right? So when the DNA is much straighter, you have the mismatch doesn't have a lot of effect, right? So all the bending data is like within a few kts of each other. So 2, 4, 6 mismatch somewhere over here will have a few kts of difference. But then as you bend the DNA even more, the mismatch effect is even enhanced for higher bending. Where over here, when you go from 2, 4, and 6, you have like tens of kts of difference. This is like one of the important things to consider when you look at the experimental data, because as you increase the force and salt, at low force and low salt, you would have a DNA end loop that's like not that highly bent, so somewhere around here, where the effect of mismatch is not that significant. But as you apply higher forces, you have a stronger bend in the plectoneme, and then from here you can see that at the stronger bend, you will have a much higher effect of DNA plectoneme, right? So again, this kind of really positively compares to our experimental comparisons, right? So to do the simulations and to compare them to experiments, I had like a 600 base pair DNA. I had a 600 and 1,000 base pair DNA molecule, right? I applied force at one end of the DNA molecule. The other end was fixed to like simulate the experimental conditions, right? So this end you can twist, and then you can twist the DNA at this end, and then you can apply the force. So I did 3 different sets of simulation, right? So there's for to compare the experiments, we did 2. Oh, sorry. One second. Sorry about that. Okay, so to compare with experiments, we did a simulation at 2 piconewtons of force and 1 molar salt concentration. So I'm going to call that high force, high salt. So this was the regime where the experiment should see plectoneme pinning, right? So And then to go into the more physiological regime, where 0.3 piconewtons of force and 0.2 molar salt concentrations, which is like a more physiological salt concentration. And I'm going to call this condition low force, low salt, right? And then to also counter this entropic effect, we did a DNA, we simulated a longer DNA molecule, because the longer the DNA molecule is, the more entropic loss you have when you stop the DNA plectoneme motion. Right. So over here, when you apply the force and twist the DNA, the simulation video, you see that the DNA absorbs some of the twist, right? And then it buckles and forms this plectoneme structure, right? And then the plectoneme structure moves along the DNA backbone. And then we are using analytics, we can track this plectoneme motion, and then we can see how much of a time it stays pinned at the mismatch. Right. So we did 2, 4, and 6 mismatches at the center of the DNA. So again, to test this ease of bending, how that affects the plectoneme pinning. And then we did multiple simulations of each conditions to have enough data to sample the entropy correctly. And then each simulation was run for around like 100 microseconds. But because with coarse grain, you kind of reducing the amount of, you kind of flatten the energy landscape almost. So the 100 microseconds of coarse grain simulation actually can observe data that experiments would take like 3 or 4 seconds to observe. Right. So our simulation are like almost the same time scale as experiments now. Right. And then show some of the preview of what the kind of things we see, right? So at high force, high salt, you have the plectoneme that's very strongly bent at the end loop, right? Then at low force, low salt, you can see that the plectoneme, even when it's at the mismatch, the bend is not as strong as a high force, high salt. Right? So the higher the bend, the more chance of plectoneme staying pinned at the mismatch. Right? And then when you negatively supercoil DNA, you kind of weaken this local structure. So you have at mismatches that are already weaker, you have this local melting. And again, this can enhance supercoiling. This can enhance plectoneme pinning at that location. Right. So to show some of the kind of traces we see in the simulations, to track the plectoneme motion, we kind of track the beginning of the plectoneme and the end of the plectoneme. Right. So that's the blue and red region here. And then the yellow center, we can see how it moves along the DNA backbone. Right. So at high force, high salt, without mismatch, you can see that the plectoneme moves along the DNA backbone somewhat. Right. But now if you have the mismatch, the plectoneme always stays pinned at the mismatch. So we have mismatches at the center of the DNA. So the plectoneme center is always at the center of the DNA. So this tells us that the plectoneme is staying pinned at the mismatch for high force, high salt. Right. And then now if you go to low force, low salt, for 0 mismatches, you have the plectoneme that moves along the DNA backbone. Right. But now if you introduce mismatches, it doesn't actually stay pinned at the mismatch, although it spends a lot of time over there. Right. So this is the kind of thing you couldn't see with experiments, where you needed the probability of pinning at the mismatch to be 100%. But through simulation, we can actually observe we can observe fractional probabilities, where even if it's not always pinned at the mismatch, you can actually observe how much of the time it stays pinned there. Right. So first we compare our simulation results to the experimental and theoretical work that was already published. Right. So over here we have a 600 base pair DNA under high force, high salt. So that's the 2 piconewtons of force and 1 molar NaCl salt concentration. That's the experimental condition here. And then you can see in experiments, there was always observed that the plectoneme was pinned at the mismatch. Right. And then in our simulation work, we observe the same thing. So over here, just to briefly go over, on the x-axis, we have the base pair step. So each base pair of the DNA is labeled some steps. So the first base pair from the bottom would be base pair 1, base pair 2, base pair 3. Because it's a 600 base pair DNA, the mismatch is somewhere at the center of the DNA, so around 300 base pair, right? And then through simulations, we're trying to understand what's the probability of the plectoneme center being at anywhere along this DNA backbone. So for 0 mismatch, so that's the blue line here, you have the probability of plectoneme center being anywhere along the DNA backbone, being pretty uniform. Right? It can be anywhere along the DNA backbone. But when you introduce the mismatch, like 2, 4, or 6, you can see that the probability of the DNA plectoneme being at the center jumps up dramatically. Right? And then at the center, we have the mismatches. The probability of plectoneme pinning at the mismatch is almost 1 for all the cases. So this again like compares favorably to the experimental observation, that the plectoneme under these conditions would always stay pinned at the mismatch. Right. And then to compare to the theoretical model of our collaborators, right, we can see that again for high force, high salt, 2, 4, 6 mismatches, the probability of pinning at the mismatch is pretty good, compares pretty favorably with the theoretical model. Right, so high force, high salt, we have good agreement between experiments, theoretical model, and our simulations. Now, as you go to more physiological conditions, so that's like more physiological salt concentration, you can see that the probability of plectoneme pinning is not that high as before, right? So for for the zero mismatch case, you have some probability of plectoneme distribution along the DNA backbone, right? For the two mismatch case, the distribution is almost similar, telling us that the two mismatch doesn't have that much of an effect of plectoneme pinning under these conditions, right? But now as you increase the number of mismatches, you see an increase in the probability of plectoneme pinning at the mismatch because it becomes easier to bend as you increase the number of mismatches. And then you can observe that even though it's like high probability of plectoneme pinning, it's not as high as the high force, high salt condition, right? And then when you compare this data to the theoretical work of our collaborators, you can see that now the agreement between theory and simulation is not that great, right? So for the two mismatches kind of close, but for the 4 and 6, it's very far off the probability of plectoneme pinning. And then for a longer DNA, where you have like a more entropic loss, you would expect this probability of spinning to go down, right? So, and we observe that with simulations. So over here for the 0, 2, and 4 mismatch case, the probability distribution of plectoneme center is almost the same, right? There is no difference. But then as you have 6 mismatches, you see an increase in the probability of plectoneme being pinned at the mismatch. So the entropic loss for a longer DNA is kind of overcoming that ease of bending that you see as you increase the number of mismatches. And now if you compare this again to the theoretical work, you can see that the comparison again isn't very great. So you have similar for 2 mismatch, but for 4 and 6 mismatch, the comparison is not very great, right? So to kind of understand where this difference comes from, we kind of also propose and start additional experiments later on, which I'll briefly introduce. But basically, just a thing to remember is at high force, high salt, you see an agreement between experiment, theory, and simulation. And at low force, low salt, you see the theory and simulations kind of diverge. And then you cannot actually study the low force, low salt through existing experimental methods. Right. So next, we look at what happens with the negatively supercoiled DNA. Right. So in your body, the DNA is always stored as a negatively supercoiled molecule. And then that's why it's really important to understand what happens to negatively supercoiled DNA. And now it's not obvious what would happen, right? So if you are negatively supercoiled DNA, you could have a scenario where the negative supercoiling weakens the structure of the mismatches so much that every time you try to twist the DNA, it just absorbs that twist by locally melting, and it doesn't actually buckle and form the plectoneme structure. If it doesn't form the plectoneme structure, obviously plectoneme pinning becomes meaningless. Or you could have a scenario where the DNA, the negative supercoiling weakens the DNA structure somewhat, right? So you have some melting, but then it's not that weak. And so every time you apply the twist, it still buckles and forms this plectoneme structure. And in this case, in this scenario, you would expect that when this happens, you would have a higher probability of pinning at the mismatch, because the mismatch, when you have it locally melted, it's kind of almost 2 single strands of DNA. So it's significantly weaker and easier to bend than a double-stranded DNA. Right? So this ease of bending should lead to a higher probability of plectoneme pinning. And because in your body DNA is always negatively supercoiled, this scenario would be ideal when the protein is trying to find the mismatch, and it'll be easy because it'll be located at one of the plectonemes, right? And to do this, again, we simulate a few different conditions, right? So over here, again, we have 2 different lengths of DNA. We limit our search to like low force, low salt regime, because as you increase the force, you kind of have this, as you increase the force, you have this scenario where the DNA becomes more and more locally melted anyway. So if you apply increasing force on negatively supercoiled DNA, you will never see plectoneme pinning. And actually you would, like, kind of between these 2 pathways, you would incentivize this pathway where at higher force, the DNA would just simply melt at the mismatch and it doesn't actually have to back out, right? So to test that also, we did like 3 different forces, increasing forces. So to increase the force, you should have this scenario where the plectoneme formation occurs less and less, right? So, and then to also test if our simulation results were somehow dependent on how much torsional stress we're applying, we also tried 2 different torsional stress values for the 1000 base pair DNA, and then we didn't see any effects, but I'll go on the data later on. Right, so again, for the negative supercoiling, we have similar where you have like different number of mismatches, and it's the simulation time and simulation method is exactly the same as the previous one. Right, so over here, we see that when you apply negative supercoiling, the plectoneme pinning at the mismatch is significantly enhanced for all the number of mismatches. So for 0 mismatch, because it's low force, low salt, the probability of plectoneme pinning at anywhere along the DNA backbone is pretty low, right? But as you increase the number of mismatches, you can see that the positive supercoiling, there's a blue curve here, is the probability of pinning for positive supercoiling is significantly lower than negative supercoiling. There's a red curve here. But negative supercoiling, the plectoneme probability of plectoneme pinning is significantly higher. So as you introduce negative supercoiling, you kind of make it easier for the plectoneme to pin at the mismatch. And then this, I'm only showing data for 600 base pair DNA, but for 1000 base pair DNA, it's also very similar, where you have this higher plectoneme pinning for negatively supercoiled DNA, consistent with the idea that negative supercoiling causes local melting at the mismatch. And then that like makes it easier to bend at the melted region and enhances plectoneme localization, right? And then next we look at what happens to the plectoneme end loop. Like again, because our theory suggests that all of this spinning is being driven by this ease of bending at the plectoneme, at the end plectoneme end loop. So that's a structure over here, right? So over here we can see, as expected, as you increase the number of mismatches for positive supercoiling, the plectoneme end loop angle becomes lower and lower because it's easier to bend. So it's 2, 4, 6, you see a decrease in the plectoneme end loop for the same force. But then as you go to negative supercoiling, you see the same effect, but it's like dramatically more bent than the positive supercoiling case. And so that's why you have like a higher pinning at the negative supercoiling condition, right? And then we also tested different forces to see what happens if you increase the force, right? And then over there we saw that the effect of force is not that significant for the 4 and 6 mismatch cases, right? It's very similar to the probability of plectoneme pinning, but you do have a significant effect for the 2 mismatch case, right? Because, and our theory is that for the 4 and 6 mismatch case, the plectoneme is already melted. So the mismatches are already locally melted. So it doesn't really matter if you increase the force, you can't cause more local melting. But for the 2 mismatch case, at lower force, the plectoneme, like the mismatch isn't always melted. So as you increase the force, you cause more local melting, and then that more local melting enhances the plectoneme pinning because it's easier to bend. So that's why you see more effect of the increase in force on the 2 mismatch case than for the 4 and 6 mismatch case. Right? And then to test our theory, we can actually look at the probability of like the amount of time the mismatches are actually intact or flipped out through our simulation data, right? So over here you have this, like if you have this locally melted structure, you can define a term what we call base flipping, right? So in an intact DNA, you have this phosphate backbone and the base group that are in line with each other, right? So both nucleotides are in line with each other. But now if it's locally melted, you would have one of the nucleotides that's kind of away from this center line, right? So that's how you can actually find the amount of time the local base pairs are actually melted by how long they stay in line or like out of line from each other, right? And then we observe that for positive supercoiling, the 2 mismatch case, right? So over here, the mismatches are at the center, so we call the base pair step 0, right? So you see that the mismatches, the filled line here is almost never locally melted for positive supercoiling. But as you increase the number of mismatches for 4 and 6, you see that you have significantly more local melting, even for positive supercoiling. And then as you go to negative supercoiling, you can see that for the 2 mismatch case, you have this very high probability of local melting, even under the same condition, right? For the 2 mismatch negative supercoiling. And then for 4 and 6 mismatch, we can see that for the negative supercoiling, the 4 and 6 are always melted throughout the simulation. So that's why it kind of explains but you don't see an effect on plectoneme pinning when you have 4 and 6 mismatches and you increase the force. But when you have 2 mismatches and you increase the force, you can see an effect of plectoneme pinning because it's not always melted, right? And then we can also, an interesting thing we can study is what happens to this plectoneme end loop structure when the bases are flipped out, right? So a thing to remember is our theory is that the protein binds to this plectoneme end loop for mismatch recognition. And the things with DNA binding protein is they like the DNA to be at a very specific angle to have very efficient binding. So the plectoneme angle here matters a lot because it kind of, the efficiency of the binding DNA binding protein binding process depends on the angle of the plectoneme. So over here, let's for negative supercoiling, let's low force. You can see that for the 4 and 6 mismatch case, you have this like single peak structure of the plectoneme end loop. Right. So throughout the simulation, you calculate the probability of the plectoneme end loop being a certain angle. Right. So you can see that for the 4 and 6 mismatch, you have this single loop distribution with the plectoneme angle. But for the 2 mismatch case, you have this like bimodal distribution, right? So where one mode corresponds to the melted 4 and 6 mismatch case, and the second mode corresponds to the intact 0 mismatch DNA, right? So we found that you can actually observe this plectoneme, this base flipping using this plectoneme end loop, because you can see a direct correlation between the base being flipped in, and being flipped out, right? And then as you increase the force, you can see, you would expect that for the 2 mismatch case, this bimodal distribution, the peak corresponding to the intact DNA goes down because it kind of, it's like intact for lesser amount of time, and the peak corresponding to the locally melted DNA actually goes up because it's more and more melted as you increase the force. And actually we do observe this thing in the simulation, where as you increase the force, the peak that corresponds to the intact DNA goes down, and then the peak that corresponds to the locally melted DNA actually goes up. Right, so we can find a direct correlation between the bending of the end loop and the base being flipped out. And then we kind of directly quantify that correlation, right? So you can see that for the 4 and 6 mismatch case, wherever for every end loop angle, the probability of the base being open is 1. But then for the 2 mismatch case, the probability of the base being open for a particular end loop angle actually decreases as the end loop angle increases. This is very interesting, something that you couldn't see with theory or experiments. Right? And then for higher forces, again, we see a similar effect. For the 4 and for the 2 mismatch, we can see a direct correlation between the end loop angle bend and the probability of the base pair being open. Right? And then finally, to kind of experimentally verify all our simulation work, we are working on a new experimental assay, right? So whereas before in the magnetic tweezer, single molecule magnetic tweezer, you only had the mismatch, we now introduce a gold nanoparticle next to the mismatch. So now, as you supercoil the DNA, you can not only observe the location of this giant magnetic bead, but you can also observe the location of this small gold nanoparticle. And using these two, you can kind of directly observe whether if the plectoneme is pinned at the mismatch. Whereas before you only had to infer it from the data, right? So over here the results would look somewhat like this, where when the plectoneme isn't formed, the gold nanoparticle and the mismatches are at some certain length, certain DNA extension, and then when the plectoneme is at the mismatch, you see a decrease in the DNA extension of this magnetic particle because it's like shorter. The DNA is shorter now. But if the plectoneme is at the mismatch, you can see that the motion of the gold particle also. So you would observe a motion of both the magnetic and the gold particle in the plectoneme is pinned at the mismatch. But then if the plectoneme is away from the mismatch, you would observe this motion of the magnetic particle, but not of the gold particle, right? And this is this is a way we can directly test our simulation results. So finally, just to briefly go over the conclusions, right? I've shown that our MD simulations could reproduce experimentally observed data at high force, high salt, right? I've shown that at low force, low salt, the probability of plectoneme being pinned at the mismatch becomes lower and less than 1. I've shown that the negative supercoiling can actually enhance plectoneme pinning at the mismatch. And because in your body, the DNA is always negatively supercoiled, this can help in mismatch recognition potentially. and we've shown that the presence of mismatch affects how easy it is to bind, how easy it is to bend at the end loop, and then how easy it is to flip out the DNA base pairs. And then we are developing new experimental methods to like rigorously test our simulation work. Right. And then with that, I want to thank my advisors, and then my lab members, and all the simulation was performed on BioWolf. That's like the NIH supercomputer. And if you're interested in any of the work I presented today, simulation work is kind of published in this NAR paper, and my work on polymers is published in these few publications. So you can take a look at them if you would like more information. I want to thank you all for listening. And if you have any questions, comments, I'm happy to address them.
Thank you, Parth, for this very interesting talk. So, like, do any of you have any question, like, any students? Okay, like, I will start maybe.
Yeah. Go
So,
for it.
when you like twisted the DNA or like applied force, like DNA, like the bending and all other things can have some kind of relaxation time. Let's say, like if you twist it very quickly, it might behave differently than if you give it enough time to relax in any particular location or this kind of things.
But I think, yeah, for in my simulation conditions, you just apply the twist almost instantaneously, and then you let,
Okay.
and then once you apply the twist, you stop, you constrain the base pairs over there so they don't rotate back, and then you let the DNA equilibrate. So the twist application is kind of less, like a few picoseconds, and then you let the DNA relax for like microseconds.
Okay.
So it's
And like,
yeah.
so in experiment, like they can't do in that way in the experiment, right?
No, they can do it. So they do the same thing in experiments where you rotate the DNA almost instantaneously. And then once it's rotated, you stop and then let the DNA equilibrate to like some DNA extension. And then once it's equilibrated, you collect the data. And then you can apply this additional turn and then stop again, collect data. So it's kind of a start-stop process. But yeah, it's not continuous.
So like if you applied this force or twist like continuously by varying just time, like I'm not sure, like the result could be like little different or something.
Yeah, it depends on. So if you apply the twist slow enough, you should get the same result.
Yeah.
But then if you apply that twist like really quickly, right, you just like directly jump into the. So if I let me go to the. So over here, if you like kind of apply the twist slowly, you should get like some similar result if it has time to equilibrate. But if you apply the twist very suddenly, so you just like apply 10 turns, which is like directly get this point, like it wouldn't matter that much, at least in the experimental sense. In the simulation sense, it'll probably be similar, right? You'll have like some fluctuation, but overall, the time scale of application shouldn't matter. It's kind of an equilibrium process, so all those things shouldn't matter.
Okay. So there is a question in the chat box. So what software did you use to model DNA, and is this cross-linked?
So I use LAMPS, and that has like a simulation parameter that has like a simulation model for the OxDNA. So I just use LAMPS. And what is the second part?
Is this cross-linked?
By cross-linked, do you mean it's like linked between here and here, or just like the backbone?
I'm not sure.
So the two strands
You
of
can just
DNA.
unmute and ask the question.
Hello, sir. So I was just asking whether adenine and thymine, is it like cross-linked, or we just able to download those models?
So Oh, so the base pairs are actually like linked by a hydrogen bond. So it's not a covalent bond. Right? Sorry, yeah, it's not actually cross-linked, it's just hydrogen bond.
And yeah, I guess like you can download the model, I guess, like the
Oh yeah,
DNA
it's part of
structure.
the LAMP package. Yeah.
I'm also working on polymer, so I have to cross-link between the monomers and different.
Oh yeah, yeah, I think LAMPs are a command for that, but I didn't use it for this one. Yeah,
Thank
this
you.
one was just like hydrogen bonds.
Any other questions? So, but I have one more question. Like you did your undergrad and like PhDs in mechanical engineering department, but
Yeah,
this works is like it feels a lots of like biophysics.
yeah.
Like
So I think yeah. So
how difficult it was for you to like switch your focus from doing all the mechanical engineering thing to DNA, learning DNA and all these biophysical things.
I mean, technically it wasn't that different because, like I mentioned, like the bead spring model got me started with like both polymer and DNA, right? And then I moved to OxDNA. It's kind of similar model, like similar coarse-grained model, slightly different. Right? So like technically it was pretty easy, but then obviously with like learning all the stuff about DNA and biology was kind of tricky. And that's kind of the most question I got about my defense was like, what about this protein, that protein? And I hadn't even heard the names of those. So those kind of challenges always like, obviously are there because like the last bio class I took was in like 9th standard or something. So I didn't know anything even till the end, but like technically it wasn't that bad. So that's something you can consider if you're trying to switch, right? Like if you can do the simulation work, it's probably work out fine, right? Because biology honestly is so vast, like no one actually knows all the things in it anyway. So yeah.
Okay, I have one more question.
Yeah.
Like how did you determine whether the local portion of DNA went into melted resin? So yeah, how do you define the melted resin?
You mean like like I use melting and base flipping interchangeably for the simulation. It's just kind of like obviously if they're in line, there's like hydrogen bond that kind of strengthens the structure. So like structurally the DNA duplex, that's like the two strands of DNA are stabilized by so the two strands of DNA are stabilized very significantly by the stacking interaction of the base. So there's a like pyrene pyrimidine rings. If you know anything about aromatic rings, right? They're like kind of easy to stack at some particular configuration. And then, so the DNA structure actually is stabilized very much by the stacking interaction of the rings and the hydrogen bond between the base pairs. So the local melting I define as the base being flipped out. So it kind of disrupts this hydrogen bonding and that kind of, so that's what I'm calling local melting in our case at least. And then I use like local melting and base flipping interchangeably. Does that make sense?
Pritam asked that question, Pritam. Okay. Do you have any more questions? Okay, so my final question is like how it's feel to work on like NIS, because we know a little bit about the NIS, but like for other parts of the world, like who are still in undergrad, like NIS, like all the COVID thing, like NIS
Yeah,
becomes a very big deal for some of us. So how's the
it's
experience
very,
there?
it was good. I mean, so like very similar to any university, I guess. Also like there's like fewer graduate students, so it's kind of a thing to consider. But like facilities wise and obviously structure wise, it's kind of very similar to any research university. So it wasn't like I didn't meet I didn't meet Dr. Fauci, so I can't know. There's nothing like different from the university experience, I guess.
So to work there, you need to have a university like affiliation. Like you have to be a like graduate student at some university, and you can collaborate, or you can like directly go PhD there.
Now you have to be a graduate student at some university. So
Okay.
like they cannot give you a PhD. So you have to be a part of a partnership program or something. Yeah, also my start was obviously like my advisor, my Dr. Das knew Dr. Newman at NIH, and kind of like I said, they wanted to work on this bead string model first. And Dr. Das had like some experience with polymer simulation. And you're obviously like close by, so they wanted to start a collaboration. And then I was just like, I was the only student in the lab who could do it. So I got lucky over there. But yeah.
Yeah. So you don't have any more questions?
Maybe I could have presented the polymer work. That would have been more interesting, I guess. For
No,
them.
this is very interesting. With lots
Oh,
of
yeah, I mean, you have to say that at the end. Just kidding.
If you don't have any more questions, then just thanks Dr. Patrake Desai anymore, and thanks for joining us and presenting your work to us. Thank you very much.
Yeah.
And if any of you have any question, like you can mail or let Kaushik or Mahmoud Islam know, and maybe I can send it to Parth.
Yeah, I
if you
think
have any
Kaushik
more
also has
questions.
my email, so if he can like forward it to people if they need, I don't mind.
Okay.
Yeah. Okay.
Okay then. Thank you very much.
Thank you, everyone. Yeah.